Today we're going to begin a discussion of molecular genetics, looking first at the structure of DNA and how DNA is replicated, or how chromosomes are replicated. And so I haven't had a chance really to talk much about the history of discovery or experiments in general, but I do want to take that opportunity today as we look at how the three-dimensional structure of DNA was determined. So as we go back into the early 20th century then, it was realized that traits were heritable, that there was a genetic component, generational component, that there was some material that was being passed from generation to generation. This accounted for the physical traits of organisms, and that this material was in the nucleus of a cell, because the nucleus of the cell determined characteristics of a cell. It was also discovered then that if you look at the nucleus, that it was composed primarily of DNA and protein. And so then the debate was, well, which of these two accounts for the characteristics of organisms? Again, leading into the early 20th century, there was some knowledge of protein structure. Proteins were known to be complex. There were thousands of proteins, thousands of characteristics. So there was kind of a natural prejudice that it must be proteins, because although the three-dimensional structure of DNA was not determined, it was known it was composed of basically 4 subunits, and people couldn't wrap their minds around the fact that how a simple molecule could code for complexity of life. So this began to change then in the late 1920s with the work done by Frederick Griffith. And he worked with a bacterium called pneumococcus, which causes a form of pneumonia, in mice. And so he was looking at disease, and there are 2 forms of this pneumococcus bacterium. There's what's known as a smooth form that has a capsule that causes pneumonia, a fatal form of pneumonia in mice. And then there's what's known as a rough form that lacks that capsule, and which is non-pathogenic or non-disease causing. And so again, he was doing some experiments, and when he introduced the smooth form to mice, they would get pneumonia, of course, and die. And when he autopsied the lungs, he would find the rough form of the bacterium in their lungs, or excuse me, the smooth form of the bacterium in their lungs. And then when he introduced them to the rough form, non-pathogenic form, they obviously would not get disease and they would live. And so again, that's pretty obvious. So then he heat killed the rough form, and as it turned out, of course, killing those bacteria mitigated their pathogenic properties, and the mice would live. And then he did a kind of an unusual thing. He took the heat-killed smooth bacterium, mixed it with live rough, and when he combined those, the mice would die. And when he looked at the lungs, what he was finding was the pathogenic form of the bacterium. So again, this kind of brings you back to 2 possibilities. So one of the possibilities is that the smooth form of the bacterium, came back to life somehow, which is not probable, or that this rough form was transformed into the smooth form, and he thought that that was a much more plausible likelihood. And he didn't know what this material was that was transforming, but he felt that some material must have been going from the smooth form to the rough form, transforming them into the smooth form. So again, he didn't know what it was. He called it transforming factor, but he thought that it was probably DNA because, as we know, and he knew at the time, proteins are very heat sensitive. So it wasn't until later, in the early, about 1944, that researchers did determine that this transforming factor was DNA. So after World War II, there was a frenzy of activity looking at DNA, trying to figure out its three-dimensional structure. And another couple of researchers, again, was looking at the issue of protein versus DNA in terms of genetic material, and these individuals were Hershey and Chase. And so they were working with viruses that attack bacteria. And when you look at the structure of these so-called bacteriophages or bacteriophages, they are composed of DNA and protein. And so the question is, what are they injecting into these bacteria when they kill bacteria? Are they injecting protein into the bacteria or DNA into the bacteria? And whatever they were injecting into the bacteria then would be the genetic material, because it changed the behavior of the cells. So anyway, what they decided to do then was work with radioactive isotopes, and they were working with radioactive isotopes of sulfur and phosphorus. So and they did that because sulfur is found in proteins, but not DNA, and phosphorus is found in DNA, but not protein. So again, they raised viruses, some exposed to radioactive sulfur, some exposed to radioactive phosphorus. They gave these viruses time to attach to bacteria and inject the genetic material into the bacteria. And then they ran the bacteria and viruses through a blender to separate whatever was on the outside from what was on the inside. And again, what they were interested in were the capsules on the outside, or excuse me, what was protein on the outside, or was protein on the inside, or DNA on the outside, or DNA on the inside. And what they ultimately determined then was that outside the cells was protein, and inside the cells was DNA. So again, the viruses had a shell of protein, and they were injecting a core of DNA into the bacteria, changing their behavior. So again, this verified that DNA must be the genetic material. And so again, this also created a flurry of activity then. And a lot of research was being done. And ultimately, Watson and Crick, James Watson and Francis Crick, are the ones that determine the three-dimensional structure of DNA, really relying on the work of many other individuals. So when we look at the Watson and Crick model, and again, it was just kind of an elegant paper, and a lot of people at the time were very jealous of them, and still maybe in some ways kind of jealous of this discovery, but it was a major discovery in biology at the time. So in the Watson and Crick model of DNA, then what we have is a backbone composed of sugar and phosphates, and then projecting from that are structures known as nitrogenous bases that we'll talk about in a second. And they determined that it wasn't just a single strand, that it was a double strand and a double helix, and that there's another strand that runs in the opposite direction. So you'll notice the orientation of the sugars is actually the opposite of this strand. And that hydrogen bonds hold the two strands together, and it forms a helical structure, a right-handed helix. And so the DNA molecule in its more stable form is a DNA double helix. Now again, Watson and Crick relied on the work of several others, and some of which I'll talk about in a minute, but one of the really crucial individuals that helped them was Rosalind Franklin, who never really got a lot of credit at the time for the discovery she did. But her work was crucial because she helped through work known as X-ray crystallography, where in this case crystals of DNA were formed, X-rays were shown through those crystals, and then that generated a pattern, photographs basically, which could be used to interpret the three-dimensional structure of, in this case, DNA. And so anyway, this famous photograph essentially is showing that, this X-like pattern is showing that it is indeed two strands, that there's a component that is perpendicular to the long axis. And so this was crucial information because people were experimenting, are there 3 strands, are there 4 strands, are there 2 strands? What's the orientation of the nitrogenous bases to the sugars and so on? So this simple photograph, seemingly simple photograph, helped answer some of those questions, and ultimately Watson and Crick took all that information and put it together the appropriate way. All right, now, again, as we look at the role of DNA then in the cell, and we're going to talk about its role in genetics ultimately, is that DNA then is a component of chromosomes. And as you may recall, we talked briefly about chromosomes before, but chromosomes are highly compact material of DNA and protein. And as we begin to uncoil that, ultimately what we're finding is that the DNA double helix wraps around histone proteins, forming these nucleosomes, and then those are packed together to actually form the chromosomes. So when we look at a chromosome, then it's composed of chromatin, a combination of DNA and protein, but the genetic component of the chromosome is the DNA double helix. So when we talk about chromosomes, a lot of times I'll talk about DNA and chromosomes, essentially as synonyms, and again, we're talking about eukaryotic chromosomes here. I'll essentially use those terms synonymously. So when I talk about DNA double helix, really, I'm talking about the genetic basis of a chromosome. And this is the material that's passed from generation to generation and determines our physical traits. Okay, so we're talking about nucleic acids. There are 2 nucleic acids, deoxyribonucleic acid and ribonucleic acid. So we're going to be focusing on DNA today. But there is another nucleic acid, RNA, that we'll talk about in a future module. Now, nucleic acids are polymers. And so the monomer unit of a nucleic acid is a nucleotide. So nucleotides are composed of a sugar, either ribose or deoxyribose. And since we're talking DNA, it will be a deoxyribose sugar, a phosphate group, and then a nitrogenous base of some type. And there are 4 different nitrogenous bases in DNA: adenine, guanine, cytosine, and thymine. And I'll talk more about those in a second. So again, what we have then is a sugar phosphate backbone with nitrogenous bases projecting out perpendicular to that. And then the second strand will run in the opposite orientation. Again, we have our sugar phosphate backbone with so-called complementary bases coming off from the side, held together with hydrogen bonds. So again, looking at the sugar then, in the case of DNA, that sugar is going to be deoxyribose. And so deoxyribose has an H here, whereas ribose has an OH here. So DNA has deoxyribose sugar, RNA has a ribose sugar, but we'll get to RNA in future modules. Now also, as we look at the structure of DNA in a little more detail then, what I want you to remember is something we've talked about before. The genetic aspect of DNA is that DNA nucleotide sequence determines the amino acid sequence of proteins. That in turn determines protein shape, that in turn determines protein function. So we want to think about that relationship because that's an important relationship and it's one I'm going to emphasize throughout this unit effectively. So this is something that's known as the central dogma. So DNA nucleotide sequence determines amino acid sequence, which determines protein shape, which determines protein function. Now, when we look at nucleotide sequence then, we need to have some kind of orientation because we're looking at a code. Essentially, it's a language. So just as in our language, English, we read from left to right, if we read letters from right to left, they don't have the same meaning as if we read from left to right. But in the chemical world, there's no real orientation. Up, down, left, and right can change constantly as particles move around. So we need some kind of chemical orientation. And so that's where the deoxyribose sugar comes in. The carbons are numbered. So we have, carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. And this is going to help orient us then when we look at the assembled strands of DNA. So if we're looking at a single strand of DNA then, what we have then is a 3' end because that's the end that's closest to the 3' carbon, or the number 3 carbon. And then as we move through nucleotides, and this could be literally millions of nucleotides, when we go to the other end, we're at the 5' end because that's closest to the 5' carbon. So again, if we look at the position of carbons, carbon 5, 4, 3, carbon 5, 4, 3, carbon 5, 4, 3, carbon 5, 4, 3, and so this becomes the 3' end, this becomes the 5' end. So that will help because, again, if we look at the sequence of nucleotides, we need to know whether we're reading from 5 to 3 or 3 to 5 because it won't have the same meaning and it won't translate to the same protein structure if we don't know the order of nucleotides. So again, as we look at the assembled DNA double helix then, again, we're going to have a 3' carbon here on this sugar. We're going to work through our strand, have a 5' carbon, and on the opposite strand, it's just the opposite. So opposite the 5' end, you will have the 3' end, and then if you go to the opposite end of the strand, you'll have your 5'. So 5', 3', and then opposite the 5' end of one strand, you'll have the 3' end because the other strand is literally running in the opposite direction. So again, we're looking here at the uncoiled DNA double helix. You always need to keep in mind that it is a helix, but we uncoil it so that we can better understand the structure. So again, always have an understanding of which end is 5', which end is 3', and then that relates to the sequence of nucleotides. Now, the last component then would be the nitrogenous bases. And so when we look at the nitrogenous bases in DNA, there are 4 nitrogenous bases: cytosine, thymine, adenine, and guanine. There is a 5th nitrogenous base. This is used in RNA, but we'll talk more about that again in the future. Now, you'll notice that nitrogenous bases then are complexes of nitrogen and carbon in either a single ring structure or double ring structure. So those that have a single ring structure we refer to as pyrimidines. Those that have a double ring structure we call purines. And so when Watson and Crick were putting their models together and trying to figure out the actual structure of DNA, what they realized that if you had 2 purines together, that would be too wide. If you had 2 pyrimidines together, that would be too narrow. So if you had a purine and a pyrimidine together, that would give you the appropriate diameter. And then they relied on the work of a researcher named Chargaff, who had determined that adenine and thymine were always in a 1-to-1 ratio in DNA, and that guanine and cytosine were always in a 1-to-1 ratio in DNA. So again, as they built their models, what they realized is that adenine will always be opposite thymine, or thymine always opposite adenine, and guanine and cytosine would always be opposite one another. And if you put them in the opposite orientations, that they would form hydrogen bonds with one another. So we describe the DNA double helix then, the 2 strands of DNA, as being complementary to one another. So again, thymine always opposite adenine, cytosine and guanine always opposite one another. So again, what Watson and Crick realized pretty quickly is that if you know the sequence of one strand, you also know the other sequence, because you'll always have that a one-to-one of thymine, adenine, guanine, cytosine. And again, held together by hydrogen bonds. So the best way to really learn the structure of DNA, quite honestly, is to draw it, and draw it in detailed structure, as detailed as you can. So if I were to give you this sequence, 5' GTA, short sequence of DNA, could you draw that? And I think you should try to draw it. And so what you would want to do is first start with your sugars. So you'd want to draw out a deoxyribose sugar, and then space out a second, and then space out a third. And then again, in the DNA structure, the sugars are linked together with phosphates. So you'd want to draw in your phosphate group linking the sugars together, forming the sugar phosphate backbone. Then, what's the 5' end? What's the 3' end? Well, again, if we look down here, here's our 3' carbon, and then if we keep going in this direction, the last carbon we get to is a 5' carbon. So here's our 5' end of our three nucleotide sequence. Here's our 3' end. And then you could go ahead and block in your nitrogenous bases. And so guanine has a double ring structure. And again, I'm just using blocks to represent that there are two rings, so we don't have to be perfectly correct here in terms of structure. Thymine is a pyrimidine, so it has a single ring structure. And adenine is another purine with a double ring structure. And then you could go ahead and put in the letters to represent those. So that's one strand. But if you know one strand, remember, you also know the sequence of the other strand. The other strand would be running in the opposite direction. So you literally then could turn your paper over and draw in your sugars, leaving space, for the nitrogenous bases. You'd link those with the phosphates as you did before. And then put in the appropriate nitrogenous bases. Again, opposite guanine, you'll always have cytosine. Opposite thymine, you'll always have adenine. Opposite adenine, you'll always have thymine. And so then you can go ahead and put in your letters. And then last but not least, just indicating hydrogen bonds. So guanine, cytosine form 3 hydrogen bonds. Adenine, thymine form 2. And then also be sure and indicate 3' and 5' ends there. So again, I think if you do that, and you could just make up any sequence you want, quite honestly, at the end of that, you really understand the structure of DNA. You understand the complementary nature of the nucleotides, you know how they fit together, and you know all of the components. So I encourage you to do that. Now, most of the time, we'll just be doing line diagrams in the future, looking at structure of DNA. So again, if I gave you an arbitrary sequence, and this is just a made-up sequence of DNA, I've shown you 3', 5', what would be the complementary basis? So hopefully you realize that, opposite guanines, and of course, again, if we have a 3' end here, if we have a 3' end here, we're going to go with a 5' end here, 5', 3'. And again, you'd start putting in complementary bases, so opposite guanine, cytosine, opposite adenine, thymine. You see thymine, adenine opposite, guanine, cytosine, cytosine, guanine. So again, any sequence you see, hopefully you can put in the opposite sequence. And then remember, 3 hydrogen bonds between guanine and cytosine, 2 between adenine and thymine. And those hydrogen bonds then are what hold those strands together. Okay, now, Watson and Crick then realized that this really shows the foundation, the probable foundation as to how chromosomes are replicated. Because again, if you have 2 strands of DNA forming a chromosome, if you separate those strands and build in complements, you would then have 2 chromosomes that are identical to one another. So again, they didn't actually end up doing this work, but they immediately realized that this was probably how chromosomes replicate, and they actually were correct about that, although others worked out the details. So the molecules that make nucleic acids are known as polymerases. So polymerase builds nucleic acid strands. And what they do is they match up complementary bases to an existing strand of DNA. And the new strands will always be built in the 5' to 3' direction. So we always want to keep directionality in mind when we talk about DNA and RNA. So there are 2 types of polymerase. DNA polymerase makes new DNA strands. RNA polymerase makes new RNA strands. And we're going to be focusing on DNA polymerase. So DNA polymerase has kind of a unique characteristic to it in that it must bind to a double strand of a nucleic acid. And again, it also builds 5 to 3. But that first characteristic is really important because that's something that is different compared to RNA polymerase. So it must bind to double strands, and then again, it will always build 5 to 3. So those are things to keep in mind. Okay, so again, kind of a simplistic view of what's going to happen here then is that what happens when chromosomes are replicated, when we need to make new chromosomes. And the reason we need to make new chromosomes is that if a cell is going to divide, it needs to make a new set of chromosomes so that at the end of the division, each cell ends up with a full complement of identical chromosomes. So you separate the strands first, and then in this case, DNA polymerase will come through and match up, take the existing strand and then match up a new nucleotide to that. And again, if you know one strand, you know the other, and vice versa. And again, the polymerase has to work in the 5' to 3' direction. So they'll physically be working in opposite directions, but their polymerases will work, again, 5 to 3, 5 to 3. So they're physically moving in opposite directions along these two strands, but chemically they're working in the same direction, 5 to 3. So at the end of replication then, and the reason we call it replication rather than duplication is that you're not taking one strand and making a new strand. At the end of this, we have what we call a semi-conservative replication. Each new chromosome will have one original strand from the original chromosome, and then one new completely strand that was synthesized based on the template of the original strand. But we do end up then with two chromosomes that are identical to the original chromosome, assuming no mistakes have been made. So, just very quickly, and I'll talk about these as we go along, some of the enzymes involved in chromosome replication include helicase, which unwinds the double helix, something called primase, which actually makes a short primer of RNA opposite a DNA strand, and we'll see that in a second. And then DNA polymerase, which will build new strands of DNA, and something called ligase, which splices fragments of DNA together. So we'll see the role of these as I go along. All right, so let's go ahead and look then at replication. Let's assume we're starting with a double helix, and I've unwound it for simplicity's sake here, and realize again that we've got 3' end, 5' end, opposite 3' end, 5' end. I'm not showing you the nucleotides here just again for the sake of simplicity. But what will happen is that there's a complex that will unwind the double helix and keep the strands separated. So helicase will unwind the DNA double helix, separate it, and there are some single-strand binding proteins that will bind to those and keep those separated. Then primase will build in short sequences of RNA. And the reason for this then is that if we go back to single strands, polymerase can't bind to a single strand. So polymerase is not going to be able to bind to this single strand and build a complement. It needs a double strand to work off of. So primase then builds a short sequence of RNA. That now allows DNA polymerase to attach, and from that point it can continue to build DNA strands. So the black lines here then represent the new strand of DNA opposite the original strand. So again, it would read the existing strand and then match up the complementary bases as it moves along. But again, keep in mind here that they have to work 5 to 3, so they're physically working in opposite directions. So as the chromosome continues to be unwound and separated then, you'll notice that this strand is already, or this polymerase is already working 5 to 3. It can keep going in that same direction, and it will. It'll keep working 5 to 3. And so we refer to this as the continuous strand, or sometimes the leading strand. And so what's happening on the other strand, however, then, is that this polymerase has gotten to the end of the line here, and it can't work in the opposite direction. So what that does is create an opportunity for another primase enzyme to come in, lay down another primer, and the circles, by the way, are representing the DNA polymerase here. DNA polymerase now can bind to that double strand and build in another section of DNA. So while one strand is being built continuously, the so-called leading strand, the other strand is being built in pieces. This is the so-called lagging strand, and these pieces are known as Okazaki fragments. So Okazaki fragment, Okazaki fragment. So as we continue this out then, again, the leading strand will just continue to build in a continuous line, whereas the other will require more primase, new polymerase, and another section being built in. And then, at the end of this, the primases, another form of DNA polymerase actually will clip out the sections of RNA, because ultimately the chromosome has to be made completely of DNA. And then another form of, again, of DNA polymerase attaches to the double strand, fills in the gaps. And then it can't actually connect the Okazaki fragments together. And so that's the role of ligase. Ligase actually finishes out this linking of the sections together, and then we have our two complete strands. So one thing I do want you to notice, though, is that at the end of the chromosomes, the so-called telomeres of the chromosomes, we will have shortening of the chromosomes. So one of the things that does happen with each round of division is that chromosomes get shorter. And some people think that this is what contributes to aging, because a lot of cells then have a finite number of divisions that they can complete before the ends of the chromosomes get too short, and at that point then chromosome replication will not be initiated. So I want to go through this one more time again, just a little more quickly then, showing you this replication of chromosomes. So again, we start with a particular type of chromosome. Helicase will separate the strands. Single strand binding proteins keep those separated. Primase will build in short segments of RNA, which allows DNA polymerase to bind, and then they build 5 to 3. And as that continues to expand, one strand is built continuously, so-called leading strand. The other side will be built in pieces, Okazaki fragments, so primase binds and builds. And then the polymerase builds rather, then one continues on, polymerase binds and builds, and then the RNA segments are removed, and the final segments are built in, and then ligase puts that all together. So we end up then with two identical chromosomes, albeit again with the ends slightly shorter. So again, this would be the first step in cell division. So you first have to replicate the chromosomes, and then the cell can go ahead and divide. But we start with one chromosome, end up with two genetically identical chromosomes. So as we look ahead then, one of the things I want to say is that DNA is just an amazing molecule. We use a lot of analogies for it. Sometimes it's described as the so-called blueprint of the cell and that kind of thing, but there's no good analogy for DNA. DNA is a remarkably unique molecule. It can replicate itself. It directs not only what characteristics a cell will have, but it also codes for the tools that are required to make itself as well as the cell itself. So again, there's no good analogy for DNA, and I'll talk more about that in future modules.